moving ahead, we have your next topic, that is plant breeding and genetic modification. Now, plant breeding will firstly involve principles and methods. That is, it will start right from the selection procedure. Which seed would you select? Why would you select it? What will be the criteria of this selection? Then, if it is not sufficient enough to suffice your ultimate demands of the product, final product, you can opt for methods of hybridization. Then there will be several approaches to hybridization. Some will be at a cellular level, at a molecular level, at a recombinant DNA level, or simply through grafting. So at various levels you will have hybridization taking place, and you have to study them with examples. There is heterosis, there is male sterility. Then moving ahead, linked to the topic of genetics, you have genetic maps and molecular markers. So these two topics are very much linked, and I would suggest you to study them in one flow. Rather than taking breaks in between, you please study genetics and plant breeding genetic modification in one flow. So that will help you generate a mental map of how these things are related to each other. You also have sporophytic and gametophytic self-incompatibility. There is haploidy, triploidy, there is somatic cell hybridization. Now, what are all of these things? These are basically the abnormal happenings or abnormal phenomena which take place in nature, or sometimes they are directed in laboratories for particular purposes. So, you will have to study their significance in such a way that if it is directed, say, in a laboratory, then what is the ultimate aim? What is the benefit derived out of it? And if you are studying this at the level of nature, if it is happening in nature, what is the impact? Is it going to help the plant in any way? Is it going to affect the nutrition or the ultimate plant's product in any way? So with that aspect in your mind, you have to study your different type of incompatibilities and variations which take place in normal fertilization and growth of plant. Going ahead, you have marker assisted selection. There is gene transfer, which can be direct and vector mediated. There is plastid transformation. Then you have transgenic plants and their application in agriculture. There is molecular farming and plantibodies. Now these all topics are directly the application base of your recombinant DNA technology with relation to botany. Now, why these topics are of concern? Because especially in India, we are an agrarian economy, and developing better generating yield, better generating plant in a given area with minimal input is what we all want. So studying these methods is one of the prime concerns of humanity, because ultimately it is going to help us generate the very basic need, that is food. So please study these methods in detail as to, if for example, say if you're studying gene transfer, then study gene transfer with relation to the different genes involved. Don't just study the procedure. Study with the help of examples which are given in your reference books. I'll tell you the name of reference books which you can refer. So when you study it in that aspect, that is how you will prepare for your exam point of view. Studying only the theory and procedure will suffice only for your board examinations or university examinations. But for competitive examination, you need to know the examples and names of genes in details. Sometimes the genes are selected from different plants, so you also need to know the sources of the plants. So with that aspect in mind, you will be studying this topic. And reference books for the same will be, there is one very simple approachable book, that is by B.D. Singh, Plant Breeding Principles and Methods. Then you have another book, that is Principles of Genetics by Gardner and Simons. There is also another book on plant breeding by Fundan Singh. So apart from these, there might be other books available in the library, as I keep repeating. So these should suffice your preparation for this particular topic.